95%. Let that number sink in. 95% of Earth's ocean is unmapped, unobserved, and unexplored. We photograph black holes millions of light years away. We land robotic explorers on the dusty red surface of Mars. We analyze the atmospheres of planets in other solar systems. Yet the largest single habitat on our own planet remains a mystery. It is a realm without sunlight, a world of crushing absolute pressure, a place of bizarre life forms that thrive in conditions that should be impossible. We call it the deep sea. For centuries, we believed it was a barren void, a lifeless desert of mud. We were wrong. It is a world teeming with life, but it is not our world. It is a world of monsters, not monsters in the fictional sense, but in the original Latin meaning of the word. Monstrum, a divine omen, a creature that defies classification. We have sent our most advanced robotics into this abyss. We have pierced the darkness with high-intensity lights, and we have returned with footage. Footage that has left scientists baffled. Creatures that do not fit our established tree of life. Sounds that should not exist. Sights that defy explanation. The central question is not what is down there. The central question is, with all our technology, all our knowledge of biology, why can't we identify them? Before we can understand why these creatures are a mystery, we must first understand where they live. The deep ocean is defined not by geography, but by physics. The primary challenge is not water, but pressure. Imagine the weight of the air around you. We call that one atmosphere. It is the pressure we have evolved to live with. Now, descend into the ocean. Every 33 feet or 10 meters, the pressure increases by another full atmosphere. The average depth of the ocean is 12,000 feet or 3,700 meters. At this depth, the pressure is over 370 times greater than at the surface. It is the equivalent of having a fully loaded jumbo jet pressing down on every single square inch of your body. Our most advanced nuclear submarines cannot survive this. They are crushed like tin cans. To explore here, we must use specially designed vehicles like the submersible Alvin, a 10-ton sphere of titanium, or more commonly, ROVs, remotely operated vehicles. These are our robotic avatars, our eyes in the deep. But pressure is only the first barrier. The second is darkness. Sunlight penetrates the ocean to a maximum of about 1,000 meters or 3,300 feet. This is the epipelagic or sunlight zone. Below this lies the mesopelagic or twilight zone. Here, the light fades to an eternal deep blue. And below that is the abyss, the bathopelagic, abyssopelagic, and hadal zones. From 1,000 meters down to the deepest point on Earth, the Mariana Trench, nearly 11,000 meters deep, there is no sunlight, none. It is a world of total, permanent darkness. And finally, there is the cold. Away from the volcanic life of hydrothermal vents, the water here hovers just above freezing, around 2 to 4 degrees Celsius. This combination, crushing pressure, total darkness, and near freezing cold, makes this environment the most extreme on Earth. It is, in almost every measurable way, more hostile to us than the surface of Mars. This is not an ecosystem we can visit. It is an ecosystem we can only observe, briefly, from behind the lens of a multi-million-dollar robot dangling on a miles-long tether in a heaving ship on the surface. This is the fundamental problem. We are aliens here, and we are seeing things that have evolved for 1 billion years in an environment we can barely survive for a few hours. Our tools for peering into this world are miracles of engineering. You have seen the footage. It comes from ROVs like Hercules, operated by the Ocean Exploration Trust, or Deep Discoverer from NOAA, the National Oceanic and Atmospheric Administration. These machines are outfitted with high-definition cameras, powerful lights, robotic arms for sampling, and containers to bring specimens back to the surface. For decades, this was our only method. Go down, look around, grab what you can, and bring it up. But this grab and retrieve method has a fundamental flaw. Many of the creatures in the deep are gelatinous. They are made of the most delicate, fragile tissues. When brought up from the crushing pressure they call home to our low-pressure world, they disintegrate. They turn into gelatinous mush, unrecognizable. We were only seeing the things robust enough to survive the journey. But now, a new tool is revolutionizing deep-sea exploration. A tool that doesn't require a camera or even a specimen. It's called eDNA, environmental DNA. Every living creature sheds DNA into its environment. Skin cells, scales, waste products. All of it floats in the water like a genetic soup. Scientists can now take a simple bottle of seawater from the deep, filter it, and sequence all the DNA fragments it contains. They run these sequences against our vast global databases of known species. And this is where the mystery deepens. Because they are finding DNA that matches nothing. No known fish, no known squid, no known whale or crustacean or bacteria. It is ghost DNA, the genetic signature of creatures we have never seen, never captured, and never classified. eDNA is telling us that the abyss is filled with a dark matter of biology, creatures whose presence we can detect, but whose form we cannot imagine. We are haunted by genetic ghosts, and we are baffled by the forms we can see. The ocean is not a silent world. It is filled with sound. And in 1997, NOAA's hydrophones, underwater microphones placed in the deep Pacific to monitor volcanic activity, detected something. It was a sound that was both powerful and bizarre. It was recorded by multiple sensors thousands of miles apart. Using this triangulation, scientists at NOAA traced its origin to a remote point in the South Pacific, west of the southern tip of South America. The sound was of an extremely low frequency, yet incredibly powerful. It was far louder than the call of any known animal, including the blue whale, the loudest animal on Earth. The acoustic signature of the sound didn't match any known man-made source, like a submarine or a bomb, and it didn't match the signature of an earthquake. It was named the bloop. For years, the bloop remained one of the greatest unsolved mysteries of the ocean. The profile of the sound was consistent with that of a living creature, but if it was an animal, it would have to be colossal, far larger than any animal we have ever known to exist. This, of course, captured the public's imagination. A creature of unimaginable size, lurking in the dark, its voice captured, but its body unseen. The mystery persisted for over a decade. But science is a patient process. As more and more hydrophones were placed and our understanding of the world's ice improved, a new theory emerged. In 2008, scientists monitoring the breakup of an ice shelf in Antarctica recorded acoustic signals that were strikingly similar to the bloop. They were ice quakes. As massive glaciers calve and break, they generate powerful, low-frequency sounds that can travel across an entire ocean basin. Today, the official scientific consensus from NOAA is that the bloop was not a monster. It was the sound of a massive ice quake, likely off the coast of Antarctica. Case closed? Perhaps. But the bloop is not the only mystery sound. There is also Julia, slow down, the whistle, and upsweep. All are powerful, unexplained sounds from the deep ocean that do not perfectly match known geologic or man-made events. While slowdown is also now attributed to Antarctic ice, others, like upsweep, remain a puzzle. It is a sound that has been recorded regularly since 1991. It is seasonal, peaking in the spring and fall, suggesting it could be biological, but we don't know. The deep sea is a symphony of noises we are only just beginning to listen to. We have identified the orchestras of whales and the percussion of ice, but we are still hearing soloists whose identity remains a profound mystery.
While sounds are tantalizing, videos are concrete. And ROV footage from the last 2 decades has provided scientists with a parade of creatures so bizarre, they are known simply as unidentified. Many of these are gelatinous. Look at this, filmed in the deep canyons off Australia. It looks like a biological satellite or a piece of abstract art. This is a siphonophore. You may have heard of their most famous cousin, the Portuguese man of war. But that is a surface creature. In the deep, they evolve into forms of staggering complexity. This is not one animal. It is a colony of thousands of specialized individuals called zooids, all working together. Some form the bell for swimming. Some are for feeding. Some are for reproduction. This dandelion animal is a perfect example of our problem. Scientists believe it's a new species of siphonophore, but they couldn't collect it. These creatures are 99% water. They are as fragile as smoke. Attempting to capture them with a robotic arm and bring them through 3,000 meters of changing pressure and temperature is hopeless. They simply disintegrate. All we have is this video. We can't sequence its DNA. We can't study its anatomy under a microscope. We can't prove it's a new species. It exists only as a series of high-definition images. Or, look at this. This was filmed in 2021 near the Mid-Atlantic Ridge. It looks like a ribbon of glass.
a living spiral. Scientists watching the feed were baffled. Was it a new type of ctenophore or comb jelly? Was it an egg sac from an unknown squid? Again, no specimen could be collected. The mystery remains. This is a constant theme. The ROV lights sweep across a creature that defies all known categories. The science team on the ship above is ecstatic. They try to sample. The creature is either too fast, too fragile, or it simply vanishes back into the darkness. These gelatinous phantoms are perhaps the most common unidentified creatures. We see them. We know they are there, but we cannot hold them. We cannot study them. They are biological ghosts haunting our video archives.
Sometimes our ROVs find creatures that are not just new. They seem to be from another planet entirely. They don't fit any known category. In 2016, the ROV Hercules was exploring a deep canyon off the coast of California. The science team was scanning the muddy bottom when they saw this. It was a small, vibrant purple orb pulsing gently. The team, broadcasting live to the world, was audibly mystified. Their conversation was a rapid-fire list of confused possibilities. Is it a tuna hit, a sea squirt? Is it a new type of plurobranch, a shellless sea slug? Is it an egg sac? It was so strange, they had to collect it. They used the ROV's slurp tool, a gentle vacuum, to suck the creature into a collection jar. On board the ship, the mystery only deepened. The creature began to unfurl. It seemed to have two distinct lobes. When scientists gently nudged it, it began to disintegrate. Years later, after extensive DNA analysis, it is believed to be a new species of velutinid, a type of snail, but one so strange it had baffled the world's experts. But even that pales in comparison to what was found in 2022. In the Caribbean, off the coast of St. Croix, a NOAA ROV was scanning the seafloor at 400 meters, and it found this. Multiple small blue blobs. They were amorphous, seemingly featureless, and covered in the sediment. The science team on the live feed was once again completely stumped. To this day, we still have no idea. No specimen was collected. This blue goo creature, or creatures, remains one of the most perfect examples of an unidentified organism. It doesn't neatly fit into any known phylum. It is just the blue goo. These are not just new species. A new species of shrimp or a new species of fish is found all the time. These are what is it creatures. They challenge our most basic understanding of how life is organized. Perhaps the most tantalizing and most terrifying mysteries are the ones we cannot see at all. This is the frontier of eDNA. As we discussed, every creature leaves a genetic trail. By sequencing this genetic soup, we can paint a picture of an ecosystem without ever seeing it. In 2018, researchers began a massive project to sequence eDNA from the deep Arctic Ocean. They were looking for known species to see how they were adapting to climate change. They found them, but they also found something else. A huge fraction of the DNA they sequenced, particularly from the deepest, darkest water samples, did not match anything in the global genetic databases. It was a massive library of ghost DNA. This isn't just one or two mystery sequences. It's thousands, genetic codes for organisms that are completely unknown to science. This discovery has been repeated all over the world, in the deep trenches off Japan, in the Antarctic, in the abyss of the Atlantic.
Everywhere we look, we find the genetic ghosts of unknown life. What are these organisms? The data suggests some could be new forms of microscopic life, bacteria, archaea, but many sequences appear to be from complex multicellular animals. Are they the source of the gelatinous phantoms we see on camera? Are they colossal creatures like the one imagined for the bloop that are too smart or too rare to be filmed? Are they creatures that live inside the mud, never emerging? We don't know. This is the scientific frontier. We are standing in a dark library, holding a book written in a language we cannot read, telling us about a world we cannot see. The eDNA is screaming that the deep ocean is vastly more diverse and vastly stranger than our cameras have led us to believe. We haven't just missed a few species. We may have missed entire branches of the tree of life. For all our technology, sometimes the mystery doesn't wait in the deep. Sometimes it washes up at our feet. They are known as globsters. Massive, amorphous, unidentified organic masses that wash ashore. They have fueled sea monster legends for centuries. The St. Augustine monster of 1896, the Tasmanian globster of 1960. In almost all these historical cases, science did eventually provide an answer. These were not plesiosaurs or giant octopuses. They were the heavily decomposed carcasses of whales. When a 50-ton whale dies, its body rots in a very specific way. The blubber and connective tissue, the collagen, can separate from the skeleton and turn into a tough, rubbery, pale mass. The skull and bones often fawn away, leaving a headless blob that is unrecognizable. So, mystery solved. Most globsters are just rotten whales. Not entirely. The most famous modern case is the Zuiyo Maru. In 1977, a Japanese fishing trawler, the Zuiyo Maru, was sailing off the coast of New Zealand. When it pulled up its nets, it brought up a 4,000-pound, 30-foot-long thing. It was decomposing. It had a long neck, a small head, and 4 large flippers. The crew was convinced it was a sea monster. The ship's captain, fearing the rotting carcass would spoil his valuable cargo of fish, ordered it thrown overboard. But not before a ship's biologist, Michihiko Yano, took measurements, sketched the creature, and crucially took samples of its fin. When the ship returned to Japan, the photos caused a national sensation. The creature looked exactly like a prehistoric plesiosaur. Could a dinosaur have survived hidden in the deep ocean? The samples were analyzed. The scientific consensus, the proteins in the tissue were most similar to a basking shark. A basking shark? How? Like whales, basking sharks decompose in a very strange way. Their gills and lower jaw are weakly attached and rot away first, leaving behind what looks like a small head and a long, thin neck, which is actually its spine. The flippers were its pectoral and pelvic fins. This seems to be the definitive answer. And yet, a shadow of doubt remains. The eyewitness accounts, the sketches, the sheer coincidence of it looking so perfectly like a prehistoric plesiosaur. While science has confidently identified most globsters as whales or sharks, the ocean is vast. The Zuio Maru carcass is a powerful reminder that the ocean can produce forms that baffle us, even when we have them in our hands. These cases, the sounds, the videos, the DNA, the carcasses, all point to one undeniable conclusion. We are discovering things faster than we can explain them. Which brings us back to our central question. In an age of artificial intelligence and genetic sequencing, why is it so hard? Why can't science just identify them?
The answer is not a single simple failure. It is a series of massive, overlapping challenges. We call it the Great Scientific Bottleneck. It's not that science is failing, it's that the problem is infinitely larger than we ever imagined.
The first and most obvious problem is the one we've touched on: the sheer difficulty of getting a specimen. Biology for 300 years has been a hands-on science. To describe a new species, you need a type specimen, a physical body you can measure, dissect, and store in a museum. But how do you get a type specimen from 4,000 meters down? We've discussed the gelatinous creatures that disintegrate, but even robust creatures suffer. They are perfectly adapted to a high-pressure world. Their cells, their enzymes, their very body structure is built to withstand thousands of pounds of pressure. When you bring them to the surface, they suffer from the opposite of the bends. It's called barotrauma. This is the blobfish. It has become a comical meme, a symbol of ugliness. But this is not what a blobfish looks like. This is what a blobfish looks like after it has been violently dragged from the deep. Its body, built to be supported by intense pressure, simply inflates. It bloats, its tissues rupturing. This is what the blobfish actually looks like in its home environment, 1,000 meters down. It is a perfectly normal-looking fish. We are quite literally destroying the very things we seek to study simply by observing them. This specimen problem is the single greatest barrier. If we can't study their physical bodies, we are left guessing from blurry videos, and a biologist cannot classify a species based on a video alone. The second problem is that deep-sea life plays by a different set of evolutionary rules. On land, or in the sunlight zone, life is driven by light, vision, colors, camouflage. In the deep, life is driven by light, but it is bioluminescence, light that the creatures make themselves. This is not just a biological party trick. It is the primary language of the deep sea. They use light to attract prey, like the famous anglerfish. They use it to startle predators, like the alarm jellies that burst with light. They use it to find mates with complex, species-specific flashes. When we retrieve a dead specimen, that light is gone. The language is gone. We are recovering a silent, dark body, when in life it was a creature of complex, living light. We are trying to understand a world of silent communicators, but all we can study are their corpses. Then, there is the problem of extreme polymorphism. Many deep-sea creatures have life stages that look nothing like each other. The larval form of an eel, for example, is a flat, translucent, leaf-like creature called a leptocephalus. For decades, scientists thought they were a completely separate species of fish. Now, imagine this in the deep sea, where we can't track an animal's life. An ROV might film a new species of transparent larval creature, which is actually just the baby of a different new species filmed 100 miles away. We are collecting scattered puzzle pieces with no idea how they fit together. We may be giving 5 different scientific names to the same animal, just at different stages of its life. It's an identification nightmare. But even if we could solve the pressure problem and the language of light problem, we would face the final bottleneck: ourselves. The deep ocean is the largest habitat on Earth, and we are discovering life at a rate that is simply overwhelming. Scientists estimate there may be 10 million species in the deep ocean that we have yet to discover. 10 million! Now, how many people on Earth are qualified to identify them? The science of taxonomy, the practice of describing and naming species, is a highly specialized dying art. There are only a few thousand active trained marine taxonomists in the entire world. A single "what is it" creature, like the purple orb, can take a team of scientists years of genetic and anatomical study to properly place on the tree of life. We do not have a discovery problem. We have a processing problem. The eDNA data is pouring in, terabytes of it, filled with ghost sequences. The ROV footage is accumulating, petabytes of it, filled with unidentified phantoms. We are, in effect, building a library of millions of books we have no time to read. We lack the human capacity to process the sheer, staggering, overwhelming volume of new life we are discovering.
So, why can't science identify these creatures? Because they are too fragile to capture. Because they are masters of a language we cannot understand. Because they change their bodies in ways we cannot predict. And because, in the end, there are millions of them, and only a few of us. This is not a failure of science. It is the beginning of its greatest challenge. For our entire history, we have looked up at the stars and called that the final frontier. We were looking in the wrong direction. The true frontier, the planet's real alien world, is not in the sky. It is in the deep. The fact that we cannot identify these creatures is not a disappointment. It is an invitation. It is a sign that our planet still holds profound mysteries, still has wonders to reveal. The darkness of the abyss does not hide monsters. It hides discovery. It hides the unknown. And in a world where we feel everything has been mapped, tweeted, and cataloged, the knowledge that 95% of our own world is still a mystery is perhaps the most exciting and most hopeful thought of all.